Hello everyone, welcome to Learn Life Science. Myself Vinita Tiwari, and in this particular video I'll be telling you regarding totipotency. Okay, so the main aim of this video is to make you aware regarding the concept of totipotency, which is very much important and which entirely forms the basis of every plant tissue culture protocol. Okay, so we will discuss regarding totipotency. It is generally a genetic ability of a cell to develop into a complete organism. First thing we are going to see is what is plant tissue culture. In plant tissue culture, the plant cells, tissues, or organs are grown in artificially prepared nutrient media and they are cultured in under aseptic and controlled environment. Okay, so in simple sense, in plant tissue culture, what do? We take an excised portion of a plant, and that excised portion is called as explant. Okay, that explant is inoculated into a culture tube containing a nutrient media with desired phytohormone. Okay, and we generally after inoculation, we incubate it under controlled condition in the culture room. For example, at desired temperature, humidity, and light intensity. Okay, so what generally happens in normal condition, what do we see? In normal condition, we see that after the fertilization of male and female gametes of a plant, there is formation of unicellular zygote, right? This is a common thing in every organism, right? There is fusion of female and male gamete and it leads to the formation of zygote. Zygote is unicellular diploid cell. This single cell is having the ability to grow, to divide, and to form the embryo. Embryo is multicellular, right? And from this embryo, the cells will again divide and differentiate, and they will finally form the seed. This seed, under particular condition, is going to germinate and it is going to give rise to the plumule, that is the shoot, and the radicle, that is the root, right? And finally we observed the entire plant on the field, right? This is a general mechanism which occurs in nature. Okay, but what happens in culture condition? So this is what we are going to see, that in culture conditions you will see, for example, if I take a carrot plant and if I excise small amount of roots from this carrot plant, after taking this excised portion, that is the explant, I will simply sterilize it and I will inoculate it in a specific media that is also known as growth media or culture media. In culture condition, what you will see, that due to the presence of different hormones as well as the different components in the media, there is induction of cells to divide and to differentiate. Okay, in simple sense, the phytohormones which are present in the media induces the cells to divide. Okay, and once the cell is induced, it undergoes division, and after division it undergoes differentiation. Okay, and differentiation causes the formation of an entire plantlet, as you can see. And once entire plantlet, means young plantlet, is observed in culture condition, it is shifted for hardening process, and from hardening, that is in greenhouse, the plant is adapted, the plant is induced for adaptation where it is properly mixed with the environment, and then it is finally exposed to the field, okay, or to the natural environment in stress conditions as well. Okay, so we will see that what actually is happening in this culture tube. So in 1962, the father of plant tissue culture, that is a Haberland, he proposed that there is a technique of development of complete plant from a single cell. Demonstrated that the somatic cell under appropriate favorable conditions can divide and differentiate to form a complete plant. He mentioned that if you take a single somatic cell and if you culture it under appropriate conditions, under proper media or phytohormone containing media, that somatic cell can divide, and that after division it can differentiate as well. and after differentiation it can form the entire plantlet with root as well as shoot and every part of the plant. Okay, so now here you can see that this potential of a cell, of a single cell, to grow and to divide into a multicellular higher organism is termed as totipotency. Totipotency in simple sense is the genetic ability of the cell to divide and differentiate into a complete organism. In this case, that organism is the entire plantlet. Okay, so again, totipotency is the total genetic potential of a cell to form a complete organism. Okay, so now we will study it with the help of a diagram. Okay, for example, you have taken a parent plant and you have taken a small portion of leaf from this plant. Okay, now this plant is a mature plant, right? And the part you have excised is also mature, right? Mature in the sense the isolated part of the leaf is mature in the sense it is in differentiated form, means it has already gained its function. Okay, so when a cell is differentiated, a cell is said to be differentiated when it has any function. Okay, means it has already performed its function, that is gained function, and it also cannot divide. Okay, and if a cell is undifferentiated, if a cell is undifferentiated, it simply means that the cell has no function attained or gained, and this undifferentiated cells can divide. Okay, it is going to divide unless and until a differentiation signal is provided to it. Okay, so now if a cell is differentiated, means it is not going to divide, and if a cell is undifferentiated, then it is going to divide. So in the previous case we have seen that the zygote was dividing and it was forming the embryo, right? It means that the zygote was undifferentiated in that condition, right? And due to its undifferentiated condition, it was having the ability to divide and differentiate laterwards. Okay, so now what we are going to do, we are simply going to convert this mature leaf. For example, I have inoculated the excised leaf portion into a culture media. Okay, you can see the leaf portion here, and due to the presence of different phytohormones, this excised portion is going to revert back to its meristematic state or undifferentiated state. Okay, now what you are going to do, you are going to induce this cell to convert back to its undifferentiated stage. Why we are converting it back, why we are reverting it back to its undifferentiated stage? Because we want this cell to divide, and for division to occur we want a cell which is in its undifferentiated form. Okay, previously this cell was mature, it was differentiated and it cannot divide. Okay, so therefore we are inducing it for undifferentiation. So the process in which this mature cell is reverting back to its immature form, this process is called as de-differentiation. This process is called as de-differentiation. Okay, once it is de-differentiated or undifferentiated, it is going to divide, and on further division, now after a particular time, there will be induction of signal by different phytohormones, and now these divided cells will gain a particular function. Gain a particular function means some of the cells will form the leaf, some will form the roots, some will form the stem, as well as other parts of the plant. Okay, now they have started gaining a function, means now they are said to be a differentiated cells. Okay, so once they gain a function, they get into its differentiated stage, they are not going to divide. Okay, so totipotency depends on these two components, that is cell division and then differentiation. Okay, so in this case we are just reverting a mature cell to its immature state, undifferentiated stage, so that it can divide. And after division, we will induce the cell to develop into a plantlet by by gaining a function, that is by getting differentiated. Okay, so this cell division and differentiation is very much important for totipotency to occur. There is also a statement that the potential of forming all types of cells lies mainly in cellular division and differentiation, as you have already seen, right? From this, it is clear that all the genes responsible for differentiation are present within the individual cell, but many of them remain inactive. In simple sense, they are trying to say, for example, this was the mature plant and this was the mature leaf. I have taken a small portion of the leaf. For example, I have zoomed out this leaf. For example, this is a single cell of the leaf. Okay. Now the genes which is present on this leaf will contain genes for the leaf part, gene for the root part, gene for the floral part. But in case of this leaf, only those genes will be expressed only those genes are expressed which is responsible for leaf formation. The other genes are also present, like the genes for root, stem, floral, fruit, but these genes are in their inactive form, and therefore the part you are seeing is differentiated and it has gained its function to form the leaf. But what happens when you culture this leaf in a culture condition? You will see that in culture condition the genes are induced, and the genes are induced by the phytohormones or other signaling components which are present in the media. In the media, what happens? The same genes will be there, but now the cells are undifferentiated, and once after division it is going to differentiate. What simply happens, all the genes are turned on, all of the genes are turned on, so that each and every cell can express any component, any part of the plant. Okay, so they are able to express the genes only when they are in culture condition. Okay, so in simple sense, you are reverting this mature cell to its meristematic state as very similar to the zygote state. Okay, I hope you are very clear with the concept, and if still you are having any doubt, you can ask me in the comment section. I will sum up everything in this slide. To express totipotency, the differentiated cell should first undergo de-differentiation and then it will undergo re-differentiation. For example, again speaking about the normal scenario, we were having a zygote which is formed by the fusion of male and female gamete. Then it forms the embryo and then it forms the complete plant. Now from this complete plant, we are going to take a portion of leaf or any segment of a plant and we are going to culture it under culture condition. In culture condition, you are going to revert this mature differentiated cell into its undifferentiated form, right? And this undifferentiated form of the cell is also called as callus. Okay. Okay, so first we will see that the process by which you have converted this differentiated cell into its undifferentiated form is known as de-differentiation. All right, it's the method which is known. Now, what is callus? So callus is an unorganized, uncontrolled proliferative mass of cell, which I will also discuss in another video. But for now, just remember that callus is the clump of cells and all of these cells are undifferentiated, means all of them is having the potential to divide and divide. Okay, once they are undifferentiated, once they have a much division, after much division they will form a clump. This clump is known as callus. Okay, now you can induce this callus to form an entire plantlet. Okay, and the process in which we are just okay, I have mentioned the definition here. The ability of the de-differentiated cell or undifferentiated cell to form a whole plant or organ is called as re-differentiation. So that is what we are doing here. We are reverting, we are just converting this callus into a state of differentiation by which it can develop its entire plantlet or can get into a complete plant. Okay, so this process of the formation of entire plantlet from this undifferentiated cell is known as re-differentiation. Re-differentiation means the differentiation is going to occur again, and therefore we are stating it as a re-differentiation. Okay, so de-differentiation was the phenomena of mature cell reverting back to its meristematic stage and forming undifferentiated callus tissue is called as de-differentiation, and the ability of undifferentiated or a de-differentiated cell to form a whole plant or organ is called re-differentiation. Okay, I hope you guys are clear with the concept. It is very simple, and I'm mentioning again that in further sessions also I will discuss about callus too. Okay. So now if you have a totipotent cell, means you have induced the cell by the de-differentiation process, and now you are having a clump of cell that is in its undifferentiated stage. Now you can induce this undifferentiated clump of cell for embryogenesis, for organogenesis, or for histogenesis. Means by using this undifferentiated cell or clump, you can use phytohormones at a particular concentration for formation of embryoids. Okay, so these embryoids are also very much similar, and it resembles the natural uh, embryo which is formed by the sexual reproduction of plants, right? But these embryos which are formed in tissue culture are known as embryoid. They will also grow in the same manner as in nature the plants grow from the embryo. Okay, but the difference is that we are inducing here the formation of embryoid, and here the phytohormones at specific concentration are used for this process. Okay, in organogenesis we induce this undifferentiated callus cell for collagenesis, means shoot formation, or you can induce it for root formation, that is rhizogenesis, or you can induce it for both. If you want an entire plantlet, you can induce it for both shoot as well as root formation, that is known as colorhizogenesis. Histogenesis means you can also use this undifferentiated cell for study of the uh, xylem and phloem differentiation. Okay, means you can also induce histogenesis for the differentiation of the vascular system. Okay, you can study the xylem and phloem by using this undifferentiated cell. Okay, so this was the main principle behind the plant tissue culture. Okay, the totipotent cell forms the basis of this plant tissue culture. In each and every protocol, the same thing is employed. Okay, the same basic principle is kept in mind, and the protocol is designed in such a manner that the phytohormones at a particular concentration will induce that undifferentiated cell and will produce your desired organ or embryoid or any type of a structure. Okay, so I hope you understood it well. If you still have any kind of doubt, you can mention in the comment section. Thank you so much.